Hi, this is Nick, and we are looking at a kind of summary of a group that was using butanol biobricks. This is from the University of Alberta, and there's a number of people here that we could look at, and there are their advisors. So they got lots of people, lots of advisors. A place called Edmonton, Canada, and it's up in that area. And again, when we do this, we'll be able to do kind of neat things like that. And again, here are pictures, people's pictures, and basically telling kind of what they do. Um, Again, a typical fashion, we see that they were going to show you the background objectives, metabolic pathways. And so basically what this group here is looking at is alternative fuels. And one of the problems that we have with alternative fuels is that things like ethanol, they compete with our food products. Biodiesel, again, the same. We're competing for oils, corn oils, and things like that from our food products. Hydrogen fuel cells, hydrogen basically in and water out is at this point kind of difficult. And then there's kind of an older technology, which is basically butanol, which is a fermentation product. So we all know about oxidative respiration, of going from glucose to carbon dioxide and getting the energy off of there. Well, fermentation is an alternative, and some organisms, rather than fermenting to ethanol, will ferment to butanol. And so this is looking at butanol. Butanol is a 1, 2, 3, 4 carbon backbone with hydrogens with a hydroxyl group on there. Again, it looks like this. Thermodynamic properties of butanol are pretty favorable in that it actually has more energy density than ethanol. And you may know that ethanol doesn't get the gas mileage that gasoline does, because quite frankly it doesn't have the energy there. And so there again is a fair amount of energy in there. We'll go down a little bit more. They did this experimental variation, so they're comparing the activity, angular speed in a motor, with a motor being supplied with butanol and one being supplied with isooctane, which is one of the ingredients in gasoline. And we see that this graph here looks pretty favorable to that. So again, butanol would be a really good fuel to use. Butanol has been around for a while. Basically, back in the old days, to get some of this particular feedstock chemical, they would ferment C-acetabutarium in butanol fermentation. Demand for acetone was greatly increased in World War II to do a number of chemicals. But again, with the increase of the petroleum industry in the 1940s, we saw a decrease in butanol fermentation, because quite basically it was easier and a little bit cheaper to get it from petroleum distillates than it was to actually ferment it. Again, in the 1990s through the present, butanol is being reinvestigated as an alternative fuel, as we could see. Again, this picture here is showing you the chromosome of Clostridium acetobactericum, which was published in 2001, and it basically shows you all the genes that are involved in this particular organism. So basically we're looking at a fermentation pathway, and this is the pathway for E. coli. So E. coli could basically take glucose, convert it to pyruvate, and then this pyruvate then can either be oxidized kind of all the way through the Krebs cycle, which is kind of depicted over here, or it could be converted into things like such as lactate or ethanol. And here we're looking for the production of butanol through an acetate intermediate. And basically what this is showing you here, that E. coli does not have those particular pathways. And so what we would like to do is to put in this metabolic pathway into E. coli, which doesn't necessarily have it. Again, E. coli convert glucose into pyruvate, which is the starting point of the pathway, but it is not the main part of it. So let's keep on going. So the design concept is to take the genes for production of butanol from Clostridium acidobutyricum and put it into E. coli, and we can optimize it there in E. coli. Once it's in E. coli, we actually would like to, or this group, sorry, would like to put it into Chlorobium tepidum. I'm losing it here. And basically this is an anaerobic photosynthetic bacterium, so it would actually be able to produce its own glucose from it can produce its own glucose from carbon dioxide and sunlight in an anaerobic area. So again, E. coli is just used as an intermediate in order to move this particular product. And so what's going on with Chlorobium tepidum is that we has a proposed butanol carbon cycle. And so what would go in Chlorobium tepidum, or CT for short, would take carbon dioxide and sunlight and produce butanol. This butanol then can be used to combust, to burn, to drive our cars, and to do work, provide energy in order to do work. And the great thing about this organism is it grows in anaerobic conditions, does not require air, so again that means that our butanol is not going to blow up, but also it means that this particular organism is not directly competing with regular glucose for our feedstocks. So again, we could grow these in photosynthetic anaerobic chambers relatively easily. And so the objectives was to transform the genes required for butanol production into E. coli, to develop butanol tolerance in E. coli, to transform these genes required for butanol production into C. tepidum, and see if they work. And so the additional butanol metabolic pathways is, again, remember in E. coli we could go from glucose through the glycolysis pathway to give us pyruvate, which then can be converted into acetyl coenzyme A. Now normally the fermentation pathway would produce an acetaldehyde and ethanol, and again, in the presence of oxygen we could go on to the tricarboxylic acid cycle, and this would release CO2. Now, an alternate pathway that we can create would be basically taking acetyl coenzyme A, and we double it to make acetylacyl coenzyme A. We convert that to 3-hydroxybutanoyl coenzyme A, crotonol coenzyme A, butanoyl coenzyme A, butanol, and basically butanol. Okay, and this requires 1, 2, 3, 4, 5, 6 particular enzymes in order to create this product. And so again, the genes for butanol pathway is have been sequenced, and we know exactly what they are, and we can PCR these out without the sequences in them that we know. So we would take the normal black backbone plasmid, and we can clone the gene for acetyl coenzyme A transferase there. And what acetyl coenzyme A acetyltransferase does is it converts 2 acetyl coenzyme A's into an acetyl coenzyme A, and this has the 1, 2, 3, 4 carbon backbones on it, which can be modified to eventually give us butanol. Again, we're just using PCR in order to do this. Again, we want to make an operon, so we'll probably they were, sorry, I'm saying we, but it's them. They were putting a promoter up over here, and they were expressing that particular messenger RNA in order to make an operon, to make all the genes in this particular pathway. The second gene would be a 3-hydroxybutyryl coenzyme dehydrogenase, and so it takes this structure right here, and it adds a hydrogen onto this ketone group over here, and then we make this particular product, which is 3-hydroxybutyryl coenzyme A. So 3-hydroxy, 1, 2, 3. Okay, 1, 2, 3 hydroxybutanol A. So there it is. Again, the third gene, which is an enzyme called anoyl-CoA hydratase, does the next pathway, and then we could add the next enzyme in this pathway, and finally the next, the fifth, and then finally, so again on this one here, alcohol dehydrogenase, we are making this particular structure right here, and then finally we get our butanol with the enzyme butanol dehydrogenase. And so this requires us having a plasmid with 1, 2, 3, 4, 5, 6 different genes in it. Again, we can put all of these under control of one particular promoter, so we could express them only certain times, maybe at times when the cell has lots and lots of glucose hanging around in order to make this product. Okay. The cloning strategy is they use commercially synthesized coding sequences. The coding sequences, they coded coding sequences into B0034 to add the ribosomal binding site. They assembled operon in order of the metabolic pathway. So the first gene was first. They inserted the operon into J61003. They insert 10500 arabinose promoter plus ARC upstream of the operon. And so this is the promoter region as well as the gene that's responsible for cloning it. So in the presence of arabinose, if you add arabinose as an inducer, it would express this particular operon, and then the plasmid class chassis compatibility is for HB101, which is the E. coli strain. And they got this in and they got it to work. And again, they tested their constructs by looking at lane number 1. Here is your chassis, here is the DNA. Lane number 2, with both of these, we get a bigger piece of DNA. Lane number 3, we get this DNA. And lane number 4, we get this. So basically, they did the PCR in order to get all of those things. They did the XBus B1 double digest in order to kind of pull those out. Insert increasing size suggests progressive building of operon. Promoter in front of the operon in an individual gene. And so again, there's a promoter in front of here, and it's going in this particular direction, that way. The construct was verified by basically sequencing and looking at it. And so in this, so here we have that protein. Here's AMP-R, primer, CAC2. So basically we have first, second, third, fourth, fifth, 1, 2, 3. Here's AMP-R, here's the origin of replication, his tag, and then here are all the particular ones that we have. There's a specific primer for each gene, and they sequenced. Now what they needed to do is to develop tolerance to butanol, and so they made a series of tests to test for butanol. So basically they mutated the cells with cells died due to ENU, ethanol nitrosourea toxicity, but some of the cells were survived and they were mutated. Those mutated cells should grow evenly throughout to everywhere except in ENU toxicity zone. It's again, it was hard to make plates with the precise butanol concentrations. In summary, what they did is they transferred the genes required for butanol production into E. coli. They created a butanol production operon. They tried to develop butanol tolerance in E. coli, but they just didn't have enough time. And again, they didn't have enough time to transform those genes required for butanol production into C-terpidium. But basically what they did is they took something that existed in nature and they put it in there. So in future work, they wanted to basically, you need to stoichiometrically control the different genes within this pathway in order to see what it's doing. And that should basically be it. They gave their acknowledgments to kind of who they work with. You see they worked with a whole bunch of different areas. They had a bunch of different consultants looking on this. We could have the same thing. And that's basically it. And they leave with a little cartoon. It's a kit I found online. I convert the car and it runs entirely on chicken manure. That's it. Thank you very much.